these are both actually maybe sponges. The brambly thing is the main target. Couple little tiny cup corals in there too.
Push in a bit for us.
Yep.
It's good, thanks.
All right, that's what I know what that is. Thank you.
Okay, you can go in.
I know what it is. Can't remember the name of it, but I believe they collected some earlier on the dive, and we certainly collected some on a previous dive. So as we've gotten down in this kind of flatter region, the obviously the abundance and of corals has dropped significantly. We've got a couple. We've lost a lot of the primnoids. We've still got some of the black corals. We've still got some sponges, but definitely a different community down here in the kind of the caldera floor. Nice big iridogorgia.
Guess the current's flowing downhill here.
Looks that way.
Pushing on the fish over there.
Is it bathygatid? Or
Yep, I think so. It's got the super long pectoral fins.
You coming down, Jade?
Copy.
this is the same one we've been seeing, right?
Not entirely sure. Could also be Gadomus. Well, the fins are going to tell us, so I think we've gotten a good enough zoom. Thank you.
All right.
There's another eel right next to it.
So one kind of important note for people watching along is you hear us throwing the Latin names around. Almost never are we actually talking about the species name. We're generally trying to get identifications between somewhere between family and genus. So we think about Linnaean taxonomy, the system in which we organize the tree of life has different levels, from species to genus to family to order to class and so on and so forth. And then there's subcategories of each of those. But identifying these things to the species level is exceedingly difficult, any of them, fish and corals and all. So we're almost always talking about the genus or family level here on the fly, and then when it comes to sitting down and annotating these things, we'll sit through a whole host of guides and look at reference material to try and make sure we get it right. And then even then, only 30% to half will get to the species level on a good day. Some areas as little as 20% can get to the actual species level from the video.
Chris, good for 20.
Bridge nav 20165.
It's definitely a trick we use to sound like we know more than we really do.
You also mentioned that you don't use a lot of common names for these, right?
They really aren't common names.
Oh, there's not, huh?
So many of the creatures down here really are only encountered by scientists, that the Latin is the only name available in a lot of ways. And in all honesty, we're kind of inventing some of the common names as we go along.
I certainly am.
Always open to suggestions.
I mean, there's an octopus that we first saw on one of my expeditions in 2016, 2015, just north of the Hawaiian Islands, that has still yet to be described. It's pretty iconic. We recognize it pretty easily, and there's been numerous sightings of it now all across the Pacific. But we don't have a Latin name for it, and it's just, and most people either calling it a ghost octopus or a Casper. But it's one of, frankly, the few ones that has a common name before it has a Latin name. And a lot of the common names are confusing, even in the deep sea. Like the term gold coral is really annoying because there are multiple groups that all are loosely referred to as gold corals, and they're wildly different. So like...
Oh, I was going to say, is that the same for black coral too?
No, black coral generally actually refers to a group that is actually the black corals. That has a taxonomic meaning. Gold corals can be chrysogorgids because they have gold-colored skeletons. They can be Cumula manamana, which is a type of zoanthid that forms an interesting quasi-skeleton thing. It's parasitic on other corals, but it also grows its own coral and is commercially fished, or used to be commercially fished for jewelry. And then you've got corals like this that are very golden color but aren't gold corals in either category.
However, you are right that black coral as a common name doesn't really make a whole lot of sense if you're not a scientist or a jeweler, I guess. They're called black corals because of their skeleton, but when they're alive, they have tissue that can be a whole variety of different colors, but they've historically been harvested for jewelry because they have this glossy black skeleton. and it refers to a pretty large group of corals. So each individual black coral family,
Yeah, because black
There's a-
coral's what,
yeah,
a
there's a
an
lot.
order, right?
I think so.
Anthopatheria.
Anthopatherians.
You could keep moving, Dan.
Yep.
Bridge nav 30165.
And then whip coral, there's a lot of different things that can have this whip morphology,
I mean, that's a
including black corals and octocorals.
Yeah, that's a growth form, or growth morphology. Speaking of black corals. One of the earlier, I guess it was at last, I think it was actually last
quick
leg.
zoom.
What's that?
Quick zoom on this one.
Yeah,
Um,
push in there for me.
right, and that'll be good enough for an ID, thanks. One of the potential new species from this expedition is the squat lobster that we collected from a black coral at the end of last leg. Our colleagues at the Smithsonian are relatively sure it is a new species, or certainly a new record for
White, thanks.
this, for the Pacific Ocean. The only thing they can come close to from the photographs is a species that lives in the North Atlantic. So it's either a massive range extension on that species or a new species here.
Sorry, Chris.
At some point when we have a minute, I'm going to want to collect some of these little small branches of white coral.
Alright.
If this is a good spot or not.
Move just ran out, so you're good to go if you want to do it.
Yeah. A little white.
Yeah, this one, this one, this one, this one, take your pick.
Roger.
So these are pretty consistently in the genus Narella, but like I was saying earlier, it's really hard to take some of these past a genus level identification from video alone. So we've been seeing this is definitely the dominant genus of this dive. So I'd like to take a sample of it to confirm the ID and get to the species level on it for this area.
It's important to remember to sample the common things sometimes. So sometimes you'll see us getting excited about something that's rare or surprising and that being of interest for a sample, but it's equally as important to get a voucher or a representative of something that's extremely common that you might not know exactly what it is. And once it gets ID'd, that can help with video annotation and all kinds of stuff.
Hi, everybody.
Welcome.
Thank you.
Guest star on the 4-day.
Guest appearance. So what do we know about this so far? Has it turned out to be as fascinating as our dreams across the center?
Not quite as fascinating as our dreams, but a really nice dive that has been probably our most corally dive yet on either expedition, or certainly up in the top one or two.
Push in there.
Our dreams went a bit off the rails when we were planning the dive. So this is still a very good dive, but the feature itself,
Jen.
it looks like a, as Brian said, a middle school diorama of a volcano.
Awesome.
So that had our hopes up a little bit for something geological going on, but
Push in a bit
lots
tighter.
of corals, which we love.
Okay.
Top of that rock craft. Any particular branch?
Not really, but if you want to, just standard 10 to 15 centimeters, but if you want to take those three, look like they'd be convenient.
Right.
So as you can see, as the arm is touching them, the polyps are retracting and facing down, which is one of the diagnostic criteria of this group. This group also is known for having extreme levels of sclerites. Yeah, sclerites.
You good there?
Yep, that's perfect. They have got so many sclerites in their tissue, they often can't fully retract, and so right, thanks. Starboard small box.
Starboard small box. Right.
What's open?
B, C, D, and E.
So anything but the forward small box.
You can push in tight on that thing while we're putting it in the box.
Got a little cup coral hiding in the background there too.
Wee cup coral.
What sample number would this be?
Sample number 70.
Thank you.
Sorry, we said,
Anything but the forward small one.
right, okay, Jaden, you should open up, please.
Copy.
Look at that clearly legible, right? Labeling there.
Seems like there's quite a bit of marine snow moving through. Has that been consistent?
There was a
Might
big
just
yellow
be
ROV
the
in the
ROV.
neighborhood.
Okay,
Landing on this rock.
got it.
Not sure.
Okay, Jaden, you can close things.
Copy.
These are so compact. I always really like this design. Or this morphology.
Caught myself here. What's going on?
Tangled up in the headphone.
Definitely not wireless. Don't know what all the post-it notes are about. Where are we? Okay, I can go wide, thank you. Are these your notes? Or Megan's notes?
Probably.
What do they say? I
I
can't
think
even
they're
read.
Rachel's notes.
Rachel.
Checks out.
And we're off. Taya, how is it going up in the video seat over there?
It's going well. I can't say I'm doing great, but I'm trying my
Doing
best.
amazing.
Oh god.
I can say she's doing great.
Yeah, no, for your second time over there, doing really good.
Thank you. I
Like,
appreciate it.
It,
Doing my best.
that is not an easy, it is a very underappreciated skill set and how hard it is to keep the iris and the focus and everything running with all the varying distances and stuff. It's not an easy job.
Yeah, and I think everybody else that does it makes it look really easy.
That's
Stress-free.
because they have a decade of practice on you.
It's true.
Got the jump.
I mean, as Erin said, she used to sit in her backyard and just sit there with her camera and just rack focus on her running dog for hours at a time practicing.
Do you take photos at home, Taya? Are you a photographer?
Very, very amateurly. Extremely poorly, but I do take a lot of videos and photos. Yeah.
Cool.
Ready to keep moving, Dan?
Yeah, I think so.
Bridge, nav 30165.
I'm going to come to your left, Jaden, so you can look at the hill there.
Copy.
Chris, can you explain for us where we are in kind of the progression of our dive and our progress so far?
Yeah, so we've mostly climbed up we've climbed up sort of into the
Right.
crater, or the remnants of this crater, and we're just now approaching the ridge. That sort of, the half-like moon-shaped ridge that sort of defines this feature. So that'll bring us up into a little bit shallower water and more rigorous terrain.
Push in a bit there for me. Just half zoom there till I land. Okay, oh, I thought it was touching. Okay, now you can push it to
This-
adjust that
The
camera a
sea
bit.
star wins for like most basic sea star. I really love this. Ask someone to draw a starfish, and this is what
You
you're
can
gonna get.
go for a tight zoom there if you want.
I'm sure it's special in its own way.
Oh, totally. Like it's got some yellow on it. Are you
I
a
think that
scale
might be
worm?
a scale worm.
Oh, cool. See? It's got a friend.
Looks like it's got holes in it. Right.
It's also walking.
Yeah, totally motoring. Brian, how does a starfish walk?
It has 2 feet. So one of the defining characteristics of echinoderms is their water vascular system, and they have a whole hydraulic pump system internally that allows them to move around using suction, water suction feet, basically. All right, thank you very much.
And go white things.
Echinoderms have some crazy biochemistry. Urchins can basically turn their muscles to stone biochemically, so if you see all the urchin spines moving around, if they get in a nice hole that they want to stay in, they can lock their muscles in a way that makes it almost impossible short of breaking the spines to get them out of a hole. There's some conjecture that, and it is conjecture, but that the sea cucumbers may actually be able to biochemically alter their density as a buoyancy control.
Whoa.
Yeah, they've got some really fascinating biochemistry in the whole phylum.
We need to leverage that. That would be cool tech for
Yeah.
ROVs.
Figure out how we can
Or
learn from that.
gliders.
Fish.
Roger.
Fish.
You can push in on fish.
Just a quick zoom. Looks like a halosaur.
Yes. The greatest transformer, halosaur.
Nice.
Definitely a halosaur, thank you.
Thank you.
It's one of the ones I can even identify.
I know, me too. I'm excited whenever I see one.
I even got it.
Yes. 10 watch bucks for everyone.
Ah, next level, zoom and focus.
So we kind of have these like pancake rocks. What's going on with these? What would form them?
Man, I am so confused by the geology of this plate and this area. I am not sure I want to even make a guess. So this mainly looks like carbonate to me, which seems kind of surprising given the very significant overall gross morphology of this feature is definitely volcanic in origin. So this could be carbonate pavement, like you would see left over from a reef, or it could be sheet flow basalt that was formed in an eruption.
That's
Ridge,
definitely the
ridge
direction
down,
I'm
another step.
leaning.
But you think sheet flow?
Doesn't really look too carbonate-ish
Yeah.
to me, other than the color.
But the color is very carbonaty color.
One more, thank you.
We've been running around with hydrochloric acid in the lab, dripping it on the rocks, because hydrochloric acid will cause carbon dioxide gas to be formed when you mix it with carbonate. So basically you drop the acid on the rock, and if it fizzes, it's a carbonate. And almost all the rock samples we've collected, once you break them open, the core rock is carbonate around here, which is surprising to me given how tectonically active this part of the world is. I'm shocked that carbonate seems to be the primary rock type we're finding. But with all the manganese encrusting and biofouling, It's hard to tell on the fly, and I am 100% not a geologist.
Could we quick zoom on that? I think it's a
Sure.
little benthic-ctenophore.
Go ahead, push it on the.
Yeah, my best guess is that
Oh, we might have to
these
land
are
for a look
sheet
at
flows
Yeah.
of basalt, and then maybe
Interesting.
there's some carbonate ooze building up from.
I think we could
Quite a
absolutely have
pointed-
another
ooh.
cruise learning just as much about the geology.
That's really cool.
I love benthic-ctenophores. They're one
What
of
are
my,
they?
they're one
So
of my favorite creatures.
Push
What are
in
we
just
looking
a
at?
bit more. See if we can get
So-
us get their little tentacles. What's that?
Actually, this is different enough. Can we collect it?
Yeah.
So this is a benthic-ctenophore,
Bridge nav, hold position.
which is, ctenophores are also known as comb jellies. It's kind of their common name. It's a whole phylum. We actually now believe it's the most basal
Go wide
of
for a
all
minute. Oh.
animals.
Land here,
So
yeah.
the last common ancestor all animals have are ctenophores. There's been a pretty raging debate for years about whether it was sponges or ctenophores, but consensus seems to be building that it's the ctenophores. And the benthic ones, and most of these swim, and there's two big phylums, two big groups, Tenaculata, and I'm spacing on the other one. And the benthic ctenophores, I believe, are tenaculatids, and they've evolved to attach to things instead of giving up their free swimming lifestyle and attach to the substrate.
How do they hold on?
I don't actually know the details of how they hold on, but there's a group at the Smithsonian led by Alan Collins who is doing a significant taxonomic revision on this group and are actively looking for samples to help better understand the phylogenetic relationships across all the benthic ctenophores.
Ctenophores are comb jellies for everybody watching along, which couldn't describe what their combs are.
So they have these rows of teens, and they are little tiny paddles. Almost think cilia, but not quite.
Push in
And they beat in the free
close.
swimming ones beat in the water, and when you to move themselves through the water. Excuse me. And when they and so they can be bioluminescent as a group, but a lot of times you see these classic videos of these swimming, and you see these iridescent ripple patterns, and that's the teens beating, and that's actually refraction,
more
not
zoom there.
bioluminescence. Even generally is what you see in the lights.
Oh, that's amazing. Look at that. Yeah, so they end up with sort of rainbow sparkles,
Yep.
which makes me happy that that's our most common animal, or the most common animal relative.
Has it got two whips? Did it
That's
suck one in? Or?
Yes. It would have two feeding tentacles, and one has either been damaged or it's been retracted.
Dr. Brian, I have a question. So will this be classified as a coral or an echinoderm, or?
Nope, it's a ctenophore. It's its own phylum.
Oh, right, okay.
The ocean's most voracious predators, the ctenophore.
All right, I think we got the video we need for science, and then just want to clip it somewhere on the branch. Would be my recommendation. And I should also say that Alan Collins' collaborator is also a PhD student at the University of Maryland who's working on the revision for this group.
Nice. Hello to that person who's going to watch our video again in the future and see this clip. You're doing great.
Steve, I assume the preference is formalin for this then, or?
To moving target. Oh, there's the other tentacle. They're coming out. Oh yeah. Get a shot of it here without the porch in the background.
You see that in the chat, Quinlan? The preservation instructions.
Just trying to get a couple seconds of video, Brian, for it
Yeah, sounds good. It's beautiful.
without me moving the camera around.
And these gelatinous organisms, the good video really can go a long way to helping, because these things do not look great once we get them in the water and in the lab. And so having a really good video is excellent. There's actually one of, I think it's the only time it's ever happened, but certainly only a couple times, a new genus of ctenophore was described by video alone, because they're so transparent, and the video of that we can get off these vehicles is so good that there was a new genus and new species description of a type of ctenophore that lives in the Atlantic based on video alone, which, like I said, I think is the first time that's ever happened.
That's really exciting.
Yeah, that was also pretty controversial because
Totally.
typically, you know, in museum collections, people like to have a voucher specimen and a DNA sample. And like when a new species is described, there's something called a type specimen, and that's the specimen from which the species is described. So if you only have video of it, there's no type specimen. So it makes it much more complicated to compare new specimens to see if it is that species. And I'm not a taxonomist, so I don't have an opinion, but it's interesting to consider the trade-offs of trying to describe things from video without vouchers, because we have so much video data,
Yeah,
and it does take a long time to describe new species and collect them. So, and we have lots of global changes happening in the ocean, and it's important to understand biodiversity as much as we can in a timely manner. So there's definitely an argument for both sides.
Yeah, definitely. And I think a lot of communities that we work with are really excited about a future of sampling that can be even lighter touch, less invasive. You know, how can we learn about these animals in place? So I think it's a really interesting conversation about how can we get enough data about something, you know, maybe a see-through creature is a good category for one.
The 3D reconstructions and scientific artist that Mike Ford partnered with for that are just amazing. Looking at the paper, it's truly beautiful. The drawings and the 3D modeling they did from the video, I'm just amazed at how pretty the results are in that paper.
That's awesome. So Brian, can you tell us, so this is attached to, is this a coral stalk? Or?
Yeah, this is an old coral stalk that has been dead for quite some time. What you see on there are hydroids, and the benthic ctenophore settled on the tip here.
Kinlan, a question for you. How do you decide which container is best for each sample?
Oh, I guess I just talk to scientists, but usually I just look at the size of it and then just determine what the best container for it to go. And I try to reserve the bigger boxes just in case we find something a bit bigger than that.
Awesome.
Like a rock.
All right, we're done. We're good, Dan. Put it away.
Right.
Awesome. So collecting this benthic ctenophore here. Can we talk a little, how do we decide what to sample? And do we know before we go?
We have, so there's kind of two approaches. There's kind of a standard approach of looking for new species, range extensions, and samples used to, voucher specimens to identify, to help with the video annotation system project. And then we also have a whole team of shore-based scientists with their own expertises that can request samples. So we, as part of the cruise planning process, there's a list of requested types of samples wanted. And then there is, so like we know that the Smithsonian team is working on trying to describe benthic ctenophores, and so we've kind of been keeping a special eye out for that. And some things are on the fly request. We got a request from the team at the Coral Reef Research Foundation in Palau yesterday.
There for me.
To collect some of the sponges we were seeing the other day.
Okay, hold that. Thanks.
That we're now on the lookout for.
Do I still have it, or is it?
We have an escape artist.
I can't tell if I have it.
Looks like you still have it.
Okay. Sorry. Panicked there.
I'm not sure you have it.
I'm not sure either. I think I don't have it. No, I don't have it.
There you go. Now you got it.
Nice catch.
Yeah, impressive. So we're collecting this one because we don't recognize it, and because benthic ctenophores are a group of collaborating scientists, are a group of animals that a collaborating group of scientists are planning to work on. So this will be something that's cataloged and sent to the Museum of Comparative Zoology, where they'll be able to, in the style of like a library book, check out a sample and conduct further science and research on it.
And this is one of those cool things where telepresence is really kind of awesome, where we can have so many different people looking at it at the same time and collaborating across different ships. So I was out on a very different project on a different ship this spring off the coast of Chile, just about literally as far away in the Pacific Ocean as you can get from where we are now. And we collected some benthic ctenophores with the intention of sharing it with the same group of researchers who were paying attention to the video on that expedition and chiming in when we saw the benthic ctenophores that were that they needed to better understand the relationships.
Yeah, super appreciative of all of our scientists ashore being here.
You can push in there for a minute. Put this in the box.
The time is 5:30 a.m. in Palau, but that means it is all kinds of hours for everyone else around the world. Hello, little crinoid.
There you go. So while he's putting it in the box, you can work on this stuff.
That one looks very awake. Ready to greet the day.
What are we going for? C?
Starboard C is good.
Say again?
A C is good.
Roger. Okay. You can open up there. Go ahead, June.
Copy.
So if you're watching on channel 3, you can see Dan actually has the manipulator arm behind, wrapped around the shoulder, wrapped backwards, sort of thinking about put your arm behind your back and putting the samples down into the sample jars there. If you're watching over on YouTube, come on over to nautiluslive.org and you can check out 3 different channel options, different views of the deep sea. And you can send in your questions. We'd love to know where you're watching from and what you're curious about. We've got a pretty awesome collection of folks tuning in right now, starting their day with us in the Pacific,
Okay,
or
can
midday
close
across the rest of the world. Hi everybody in the UK and Germany, in Italy and France, and Singapore, Australia, Fiji, Canada, the United States, Argentina.
All right, what was the sample number, Quinlan?
The sample number 71.
All right, 71, and that's Platycyntidia.
Roger. Got a minute to look around on the rock if you want to see anything in particular.
Let's look at the, no, actually I did get a good look at that already.
Maybe there's something down low.
Dan, the people want to know, how long is the tether between the two vehicles?
30 meters,
30 meters.
give or take.
How do you pick that number?
It's just a comfortable number to let Hercules have enough space to run around, but be close enough where we can see it with Atalanta. It's an easy to manage number where it doesn't, it's not too long to get tangled up.
Awesome. Thanks for all the shout outs, everybody. Hi,
Push in there on the
Scotland. Hi, Colorado.
sponge down there, maybe.
Got a shout out from astronomy professor in Southern California. Found us on Instagram, and it's a new favorite thing to watch and listen to while you grade. Anything that helps make the grading easier. That's a compact
Sounds
little
good,
sponge.
Bridge. Thanks.
Okay, can go ahead. Oh, that's right. I forgot to push the camera out.
All right, I've got one. Anybody else have a favorite fossil that we've ever found?
You ready to get moving again, Dan?
I'm probably
Yeah.
partial to
Bridge,
beaked whale
bridge
jaw.
nav.
Oh, yeah.
30165.
There was the giant shark tooth.
Yes, Megalodon tooth.
Megalodon. That was
Yep.
so cool.
That is a good one. That's a very good one. Especially because it was so sneaky. Like, I looked at that sample. I was on board for that exhibition. I physically looked at that sample when it came up. It was a scoop of small rocks. Did not see it at all. And after the sample got sent back to the University of Rhode Island's Marine Geological Samples Lab, where they're processed, a team that was there processing them just kind of like
Right.
was doing some washing of the rocks and some like scraping, and then noticed this like very triangular shape that seemed really off, and continued to scrape, and out came this big, like
okay.
bigger than your palm Megalodon tooth. It was so cool.
So awesome.
If you want to check that out, check out that gallery on Nautilus Live.
That's good, Dan.
Right.
You can search Megalodon and you'll find a few different videos from the team recapping that. The beaked whale skulls were really cool too, Brian. I think what blew my mind there is definitely how many of them there were,
Yeah. Agreed.
We just continued to find more, and, it was really this, like, I don't know, moment for me to realize that this had been, that island had been Johnston Atoll, a hotspot of biodiversity in the deep ocean, had clearly been a hotspot of biodiversity for a very long time.
Yep. Don't expect to just find fossils just laying exposed on the ground. I always think about chiseling them out of rock or digging them out, which is what you do on land. But no, we were finding the fossils just laying on the ground, nothing covering them.
Just a little manganese frosting.
Fossils are particularly special finds in the deep sea because it is extremely inaccessible. Not really the kind of place you can do a normal paleontology survey.
Yeah.
And on top of that, the seafloor is constantly getting eaten away by plate tectonics. So the oldest seafloor is around 200 million years old. So the deep sea fossil record can theoretically only go back that far.
Yeah, good point.
Just a quick zoom. We've seen this
Yeah,
one before, I
push
think.
Push in a little for me. And a little bit more.
Yep.
That's good. Hold that for a second. Get
Very
some
cool.
video here.
Do you feel like you're like recognizing friends at this point, Harold? Folks you've seen before?
My old friends, yeah. Some familiar faces. Got a
That
little
one-
isopod
Yeah,
on
it's an isopod. Oh,
Oh.
bye. Bye,
See ya.
friend.
Okay, you can
See
go
ya.
away, thanks.
Awesome. This dive has about 5 more hours planned in it, so tune in. Stay with us. We're approaching the edge of this caldera rim, which is going to be really cool and go nearly vertical, so I'm anticipating some awesome stuff coming up ahead. Hey Jaden, you want to take this one? Why are the two vehicles tethered together? Do you want to talk to us about how our operations work in that way?
Yeah, so our ROVs are a little different. There's a two-body system, so Atalanta and Hercules. Atalanta is connected directly to the ship, and the reason for that is that as the boat experiences heave, so that up and down movement in the waves, Atalanta takes that force so that Hercules can stay pretty stable on the bottom. And so while Atalanta is doing that, Hercules is attached to Atalanta for communication and power and everything via that tether that connects them.
Awesome. It also gives us this really nice helicopter view that's on channel 2 right now, which is, I think, helps a lot with scale sometimes to understand how large of a feature we're sitting in front of, whether that's a massive
Bridge
10-foot tall
nav, another step.
coral colony, or if it's a big rock cliff. Hercules is like somewhere between the size of a Jeep and a minivan, to give you the right reference, and so can really help understand that overall size. Love it. Lots of students writing in, folks that are landlocked far from the coast are really glad to be able to bring you here to Palau, to the deep ocean. Thanks for being here with us. In this combo, these vehicles can dive to, how deep, everybody?
4,000.
4,000 meters. Yes, thanks, Dan. 40 football fields, 13,000 feet. If you paired Atalanta up with another ROV we have, Little Hercules, could dive to 6,000 meters, 19,500 feet, 60 football fields, however you like to measure. But Little Hercules is visual inspection only, no arms, so no samples, no sensors, just cameras. So different tools from the toolbox depending on what you want to accomplish and where you want to go.
Another piece of wood there.
Yeah, looks
Push
like
in on the...
Little Hercules was actually the first ROV I ever worked with.
Oh, what year was that in?
2010.
Oh, amazing. Have you given it a hug on
I
the
have
back deck?
actually. I have actually. Yes.
Oh, good.
Quinlan, they collected a piece of wood earlier on this dive, right? One of the earlier samples was wood.
I think that was right before I went to bed.
Yeah.
Yeah, I was going to say.
Yes, they did.
All right.
Sorry.
Okay,
Thank you.
good.
Okay, you can go away, thanks.
I was going to say this one's nice and collectible, but we already have one.
It's beautiful. What's up, University of Wisconsin Whitewater? Thanks for watching.
If we collect enough wood, we can have a little bonfire and make s'mores.
No. No.
Expensive bonfire.
Yeah.
The best kind.
Bunch of bryozoans under the ledge, anthomastus, getting the starting to get the primnoids back. A couple encrusting sponges. We're
Yeah.
starting to the bathymetry starting to steepen up again as we approach the crater rim wall.
This is exciting. So why do we get excited about steep, Brian? Or anyone? Back row? Front row?
Well, Dan likes to fly in steeper terrain,
Uh-huh.
but generally the steeper the terrain, there's two reasons. One is in steeper terrain you have more hard grounds exposed, so more likely to have rocks that corals and sponges can attach to. Here we already have plenty of that, and so that's not necessarily the case for excitement. In this case, it's probably getting up in the current and getting more reliable current flow, which in theory then brings more nutrients or more fixed carbon that the filter feeders can grab out of the water.
Yeah, if you make a living, if you have the life plan that you can only eat things that you can catch from the water column, it is good for you to be in places where more food is going by, or more current is going by. And we think there's an upper limit to that. It is true to have places where there's too much, whether there's physical stress from the current going by, or I don't know, I guess maybe the physics of the water going by, the velocities are too fast.
Yeah, it could be it's just hard to literally stand up again. It could be hard that it's hard to settle when you're a larvae to find the rock and attach. It could be that the water's literally moving too fast for you to catch the particles out of the water. So there's definitely a series of possibilities that we really don't know too well. I've heard of a couple plans thinking about 3D printing corals and putting them in flow chambers and stuff to get a better sense of what the different shapes affect the flow around them, which is an excellent idea, and I have thought about doing something similar.
Yeah, that's a fellow UH grad student working on that.
So.
I 3D printed it with I bought a resin printer last year on Black Friday and 3D printed a shipwreck benchy.
You got
model.
A resin printer? That's cool.
Yeah, they're really cheap now. They're less than $200. But
Wow.
the 3D benchy's kind of like a little boat you print when you first get your printer. It's a thing. And I found this shipwreck benchy, so it has all these corals growing all over it. And the detail that the resin printer was just absolutely amazing. Way, way finer than the
That's awesome.
FDM printer.
Dan's got all the toys. And speaking of shipwreck, thanks everybody for all the questions coming in. We absolutely have found and explored shipwrecks before. If you're interested in that, check out the gallery tab on All Is Live, or there's a whole bunch of playlists on YouTube as well. The deep sea has more history than every museum in the world combined. So when we come to places to explore what's here, we understand that we could be exploring geology, biology, archaeology, chemistry, ecology, physical oceanography like the currents and waves. There's a lot to understand about a place, and that includes its history. We've been mapping a lot during this expedition, so always on the lookout. Palau does have a really rich history of seafaring, anywhere from Micronesian voyaging throughout the islands, and some really famous legends of stone money lost on some of those voyages to the deep sea, up to more of a modern era. The World War II history of this region is complex with a lot of things lost. So you never know. You never know what we might find. Awesome. Some people have checked out our 3D printing models on Sketchfab and on our education resources. If you check out the education tab, there's over 150 different lessons, activities, creative challenges on the website, including a whole gallery of undersea sea mounts and the vehicle models that you can print. Okay, Brian, how big is little Hercules compared to regular Hercules?
Substantially smaller. Little Herc's like, I don't know, a meter by a meter by a meter and a half?
Like a Costco shopping cart.
Yeah, a big Costco shopping cart.
Little
Or like-
Hercules is 1,000 pounds and Hercules is 6,000 pounds.
There you go. That's the stats we need. Yeah, little Hercules got taller. It has some boosters on the bottom of Freedom Edition.
Yeah, we put some new shoes on it.
New shoes, I love it.
When's the last time little Herc went in the water?
Yeah.
Let's see. We had big plans to dive it last year in September, and we ended up needing to do some work on the junction box at that point. That was the Battle of Midway and our aircraft carrier dives. So it's had a lot of work since then, but we dove it shakedown that year. So start of the season last year. So yeah, don't regularly switch between the two. Yes, Dan, there's a question, do you have a preference between the two? But I guess I'd caveat that we don't normally like switch back and forth a lot. Hercules is definitely the primary.
Bridge nav, another 20 meters.
But do you want to go on the record, Dan, with a controversial take of saying little Hercules is your favorite?
I've only got to operate little Hercules maybe 3 or 4 times.
Okay.
I was very excited when it first showed up. I saw it in San Pedro in 2020, I guess. No, we had it in Gulfport before that.
Yeah, that's true. It was there.
Yeah, so it was before we were in Gulfport in 2020, so it must have been like 2018 and 2019, something like that. But yeah, it's very shiny, very cute. It would be really cool to have them both in the water at the same time.
Yes, indeed.
Like these shallow dives, I could put it on make it a free flyer and kick it over the side.
That'd be cool. Jaden, we got a follow-up question on your excellent two-body explanation. So folks want to know if Atalanta has a heave compensator, or is Atalanta a heave compensator?
So Atalanta is the heave compensator. It's attached to the boat with the winch. So as the boat moves, Atalanta moves. And then I'm over here controlling the payout of the winch. So how much of the length is out there, what depth Atalanta's at because of that. And that compensates for the heave that Hercules would be experiencing otherwise.
Thank you so much. Yeah, we have 21 participants in our Scientists Ashore Network portal right now, signed on. Super awesome having you with us. All these folks bring different experiences. If we were diving on archaeology-focused dives, or if we were to surprise find some archaeology, we do have several hundred people in the network overall, so we have folks that have agreed to lend their expertise and be part of our overall mission of exploring the ocean. And so there are folks we could call and get on the phone or get on our V-Link system where they could be talking from wherever they are in the world in the same way and joining the same audio conversation just from home. That's the same way Iqlal Oto, the chief researcher at the Palau International Coral Reef Center and one of the co-lead scientists for this expedition, has been joining us on some of the dives. So really leverage that to be able to bring new expertise or different expertise onto ship, especially if we get surprised and find something we're not anticipating. What are those?
Could be a dead sponge.
Or where's the wanted poster?
Push in a bit.
Yeah, we do have the sponge we're looking for.
Yeah.
That doesn't look quite the same, but let's check.
Oh, critter hiding under there. See all beady eyes.
Pneumatic or sinus shrimp hiding in
Yeah,
there.
little shrimp.
Yeah, so these are sponges. I do
Push
not
in
believe
some
they are
more
the
if you got
sponges
it.
we are looking for.
These are not the sponges you are looking for. The Jedi
So there's a little
exploration trick.
nematocarcinus shrimp
Yeah.
peeking out.
So what are the things that look like hairs projecting off?
So those are its antennas and its legs. They're
Oh,
both,
wait.
both very long.
I meant on the sponge, but excellent on the
Oh,
shrimp.
Fair enough.
Its antennas
So-
and legs are long. Tell me more.
Yeah.
That
mean-
last little joint, is that its foot? The itty bitty?
Yeah.
Okay.
So they're just very long, and it allows the shrimp to sense its environment at a distance, essentially. So it can find things with those long antennas that are a bit further away than its body.
Okay.
But the sponge is also sort of bushy looking, possibly because of just detritus buildup on its sclerites, or possibly hydrozoans growing on it.
Great, we're good, Dan, thanks.
Okay, go away, thanks.
I'm looking at the map, and we're getting tantalizingly close
I
to
know.
this
Sneaking up.
massive ridge.
We're starting to pick up
Sneaking up.
this dead coral skeleton rubble that we've been seeing at this depth pretty much on many of the seamounts we've visited.
That one looks kind of like a turtle. It's amazing.
Yeah, a little patch of Narella on the turtle rock.
Bridge nav, another 20 meters.
Do we zoom
Can we look at
shrimp,
the big
please?
shrimp?
Yeah, zoom there. Go ahead.
Big
push
shrimp.
in.
It's an oxymoron.
That's a pretty good sized shrimp though. It's almost 15 centimeters long.
Yeah, so it's got these sort of feather-like legs coming out the side of it that it normally uses to swim through the water column, as it is about to demonstrate.
This is the second one we've seen holding still.
Go wide
this
there
leg,
so I can
which is unusual.
get closer.
It's kind of rare. These things are always on the move. And seeing them sitting still is a little bit strange. But this is the second one, I think, in 2 dives we've seen just chilling.
That's awesome. They're so impressive. You know, like it would take 2 hands to hold that animal.
A good shot of its face would be helpful.
Especially the rostrum, which is the pointy part. Probably some kind of Aristeid.
All right, we can let it swim out.
Roger.
Thank you.
Okay, go wide for me. Why don't you stop there for a minute, Chris?
Roger. Bridge nav, hold position.
You have to come up, dude.
Copy.
Crab, please.
Roger.
You got time?
Push in about halfway for me there. Hey, where are you going?
So this is Chaseon microniscus. It's a crab that we saw quite a lot on our baited camera survey in the Southwest Islands earlier this year with National Geographic. Really on the move. And this was a taxa that we found in our preliminary analysis that
Zoom there. I'm way
seems
up
to
in the
show up
water
more here in the Southwest than in the main islands,
We
which
don't
is an
need a
interesting
particularly tight one.
biogeographic pattern. And I think we got a good enough view on it.
column. Right. Okay, you can go wide.
It really does not want to hang out.
Come up a bit more, Jaden.
Copy.
And the Norella really love this place.
Atalanta should be pretty much stopped.
Around it.
Okay, now you can unmute. Very good.
the Stickopathies. It's been 100 meters or so since we saw one of those. Maybe not 100 meters, but in a few minutes.
It looks like we're beginning our climb up the ridge,
Indeed.
or the truly steep part of it, at least.
Morning, everyone. This is Amanda, standing in for Kim in the Science Communication Fellow Chair. We have a question from one of our viewers. At this depth, how much water pressure are these organisms here dealing with compared to us at the surface?
Oh, I have to do math.
Yeah,
roughly in feet, about half the depth.
Who thinks in feet?
Yeah, so
People who can't do math.
what is it? So it's about 10 meters per atmosphere.
Yeah.
And we are at 774 meters. So 77 or so atmospheres of pressure.
Would that crush me like a grape under my foot, or like... Do we have any visuals?
If you translate that to feet and PSI, people can.
It's 11,000 psi.
Ouch.
Yeah, I mean,
Or 1,100 psi. Sorry, 1,100 psi.
So ouch.
if you were a diver and you had all the proper equipment and everything, you could get down there. It would take a long time to come back up. I don't remember what the depth record for scuba is off the
It's
top
about
of my head.
1,000 feet.
1,000 feet. Okay, so this is quite a bit deeper. But yeah, there's a lot of problems that happen when you go deep as a human.
Ready for 15?
Land-dwelling animal.
Good for 20, yeah, maybe we'll do a little one. See what happens. 15, yeah.
Bridge nav, 15 meters, 170.
Thank
And
you for that.
No,
People
not quite,
tend to think about it as being crushed
I'm not
by
quite lined
pressure
up.
like a can or something, but it's a little bit more nuanced than that, because a lot of pretty much all the tissue and cells in your body are fluid, so
Bring your
they
head to
aren't
the right.
compressible by water. It's really the gas chambers in your body
yeah, whatever
that
is
get
easier for you.
compressed, and the changing
Bring your head to the right a bit. We'll square
state
up
of
on the
matter of your
curve here.
cell membranes and your nerve potentials get affected. So there's a whole bunch of different physiological effects that happen under pressure. So it's a bit more complicated than simply getting crushed by a can, which is what you tend to hear about when you hear about scary things in the deep sea. But yeah, as we can see, there's a lot of animals here that are thriving despite the pressure. So while it may be unfamiliar and inhospitable to us, the deep sea is by far the largest volume of habitat space on Earth. So it is really the norm for our planet. So it's us who live in the extreme conditions.
Oh, that's interesting to think about. Thank you for that insight.
Yeah, we're looking at something like 95% of the habitable space on the planet right now.
Oh. Yeah, I love that shift in perspective. Thank you.
All right. Push in there a little for me.
Different crinoid than we've been seeing mostly. that's good, thank you.
Okay.
Quick zoom on fish.
It's like a heterocarpus shrimp. There's an urchin over on the right. You could take a quick zoom, please.
Sure. Yeah, you can push in there. That's good, thanks.
That's probably all we need, thanks.
Alright.
Actually, and we look at there's a couple things quickly here. Over here, there's some sea cucumbers.
Roger.
A tiny little sea cucumber.
And then I think there's actually a soulless sea cucumber a little bit further to the left. There, yep. So these pink things are actually also sea cucumbers, but sessile sea cucumbers that don't move. All right, thank you.
Earlier on our expedition, can we quick look at this fish? Earlier on the expedition, we found a mystery animal that was transparent and it had a really, really strange body form, and it pretty much stumped everyone, but our best guess is that it's a type of soulless, but we really don't know. There's a little rat tail. Interesting coloration on the head. Alright.
Okay.
I don't think he's going to move much for us, so
You can go wide.
thanks.
I have a question for you, Harold.
Sure.
Have you ever wanted to eat a deep sea fish?
Well, I'm a vegetarian, so no.
I didn't know that. Interesting.
There are some deep sea fish that are commercially harvested, though.
Oh, they're 1.5, Dan?
Like which ones?
No, I think we're good for
Well,
now.
some
Okay.
rat tails are. Not that species we just saw. Jeff could probably speak to what they actually taste like. Famously, the orange roughy, which is a type of deep sea fish,
Which we saw last dive.
which we saw last dive, that lives extremely long and was fished very unsustainably for a while. There's also deep water snappers, which are called saboose here in Palau. Those are eaten.
Thank you for that.
Yeah, can we look at these sponges in the bottom left?
Sure.
We're looking at our wanted poster, trying to figure out if it's the same.
It's definitely not these. We still should look at these because they're new, but
Yeah,
that's
push
not
in
the
there
one.
for 'em.
That's not the one that we got the request for.
Yeah, you can push in a bit more if you want.
All right, thanks.
Okay.
It's another Oreo. Some more of this very neon yellow encrusting sponge. Starting to pick up some Flipptrophora as well, in addition to the Narella. Oxygen saturation is climbing quite quickly as we move up this, as we move last 30 minutes or so. So Amanda, as we're thinking about kind of beginning to wrap up this cruise, what's on your at-sea bucket list that you still want to do in the next 3 days?
At sea bucket list. That's a really good question. I mean, as far as things that I would love to see, I really want to see a jellyfish. I saw, I think it was a squid the other day on the 12 to 4 watch while I was subbing in for Blodock, and that was like a dream come true. It was so graceful. It was amazing. I've loved squids since I was in high school when I had purchased a squid hat and wore it on Spirit Day. So it was a momentous moment for me. Let's see.
You should have seen my Halloween costume.
What was your Halloween costume?
It was a crocheted octopus that Megan Putz made.
Oh, that's
as a hat.
amazing. That's cute. I think I'd like to see some of the specimens in our data lab. And yeah.
Let me know when you're ready for a move, man.
Roger. That seems to be
Thank you for that question.
holding pretty good.
Is there anything that
Maybe
you'd like
sneak
to
10
experience?
closer. You want
I
it?
don't have a particular,
Okay.
one particular
Bridge
thing.
nav, 10 meters, 170.
I would love to see a Nautilus, but I think that given the depths of our dives, this will be our
Push in
last
there,
chance.
force video.
Mm. How far do they go?
About 700 meters. So we're getting right into the extreme range of their depth range right now. All right, that's good. Thank you.
Okay, go away.
That would be exciting. Have we ever, in the history of, ever, seen a Nautilus on our streams?
Huh.
Not that I know of.
Yeah, I don't know.
I don't know.
Megan would be able to give you a more definitive answer,
Yeah.
but I'm sure there'd be highlights of it if we did.
Yeah.
That would be exciting.
Yeah, I feel like I would have heard about the Nautilus by now.
Yeah, that's what I was thinking.
We do have a Nautilus shell that was gifted to us by Elle from the previous expedition.
Yeah. Okeana
Very cool.
specifically went looking for them once in American Samoa with the ROV, but did not find one.
Mm.
National Geographic had a very small ROV, and they dove it down to about 300 to 600 meters, and they were able to film a few.
Oh, that's amazing.
So the species here is Nautilus bellauensis, which is named for Palau. And it's one of the only species endemic to Palau, so only found here.
Oh, that's cool.
Looks like it. So, I don't know, I can't tell. I think we're good for another 10.
Yep. Bridge nav, another 10.
So we're just getting into pretty much vertical terrain here, or will be very shortly, most likely, as we're coming up this super steep crater wall. Got another header carper shrimp in there, and an Opsamia, mainly Norella.
Sea urchin hanging out on a coral.
Oh yeah, can we look at that?
Yep.
I missed that.
Yeah, right there. Push in about halfway for me as I come closer.
Push the coral livery button.
Okay.
Really
Push it
heavily
a bit more
encrusted
for us.
wall.
Nice
Yeah.
and slow.
Definitely increasing the abundance quite a bit of the small stuff. Looks like lots of little sponges, 3 or 4 different types.
Maybe some brachiopods.
Maybe some brachiopods, maybe some bryozoans. So this is an urchin, most likely eating this Norella. They are coral predators sometimes. All right, thank you very much.
Okay.
I got a bottle brush Chrysogorgia. It's one of the first of those we've seen this dive, or since it became a watch. My attempt to watch most of this dive failed when I fell asleep. Here, please, one of the yellow ones. I'm getting starting to pick up the paramurcids.
Push in there a bit.
It's actually probably a Canthogorgia. All right,
Go
thank you.
You good with that? We can go
I
tighter.
think if you got tighter, I'll take it.
Yeah, we can go tighter.
All right, thank you.
Okay, go in.
Oh, Brian, I have a question for you.
Sure.
Which do you think kind of came first, deep sea corals or shallow water corals?
Generally, we think shallow water corals, but then it's not a definitive answer.
How do you think, do you have any theories on how that might have occurred, how they traveled to the deep sea?
There's some really good work that's been done on that, and it's been a long time since I read it, so would like to defer until I could go do some more research.
Oh, thank you.
Because there's definitely been some really good scholarship on that, but not remembering the details of it. Can we look at the sponge piece?
Roger. Push in a little for us there.
These pictures are not good.
That's good.
All right, thank you.
Okay. Go white.
Lots of little stuff on this wall.
Oh, this is going to be fun to annotate.
Harold's like, not it.
Poor Megan is probably going to do this one. If we go up just a little bit, I see a sea spider on Thurber cam.
Roger.
Good eye.
So this is a pycnogonid, a sea spider.
Push in
It's
a
a
bit
type
there
of
on the spider.
arthropod. It's not truly a spider, but it is closer to a spider than it is to a crab or a shrimp.
I've certainly seen these eating, but I feel like I see them not eating more often than I see them eating, which I think surprises me a little bit. It's not like there's a shortage of things around here to eat.
Yeah, so they actually eat
Probably go
cnidarians,
tight on that.
so corals, anemones, and they use that proboscis to basically suck out all the juice from the polyps. It's a pretty
That's-
cool animal.
That's why we like cliffs there. Easy parking.
All right, thank you.
Okay, I can go white.
This is, oh, can we look at one of these guys?
One of the squatties.
Yeah. Yep.
Picked a static one here. Could push in a bit there again.
Yeah, this is really becoming a game of I Spy. It's another type of squat lobster.
If I can hold still, I should be able to get a tight shot of it.
Maybe a Munida. That should be a good enough image.
And we'll
Thank
go
you.
for a just a second of video here. Is that all your
Yep.
Zoom, is it?
That's nice. You can see the rostrum and the mouth parts.
So what do they eat?
So they pick things out of the substrate, small invertebrates, and they're also scavengers. So we saw these on the baited camera survey.
We saw one eating a brittle star arm. Yeah. I think
Nice.
last leg.
And the little yellow things on it.
Yeah, it's got these like
Okay, you can go away.
hairs on its front appendages.
That one looked like its rostrum was kind of broken.
Yeah, that's what it looked like.
So we just got a question for our ROV pilots. Do you have a favorite feature of the ROVs?
You got a favorite yet, Jade?
I would definitely say the craft arm is my favorite feature.
Mine too.
Cool. What do you like about it?
Well, Dan, is there an official term for the control, spatial controlling of it?
It's 6 degree of freedom, 7 function, spatially correspondent.
Yeah, so basically when we're controlling it.
Sorry, I forgot the teleoperated.
Yeah, we have this basically a mini version of the arm that we move around up here to control the different joints on it. So getting to use that is pretty fun.
Oh, that's awesome. Thank you for that.
You gotta look at that dead sponge thing there.
Yeah, we'll get a tight shot here for bonus squatty there. Okay, you can push in there on the, should be able to push all the way in if you want.
That's good, thanks.
Okay, can go away. The fun part about the vertical too, we can get the camera a lot closer to what we're looking at. Stick it right up to your eyeball.
Got a big fish around the corner. There it is.
I don't see him.
Dead center top. Just went out of frame.
Oh, I see it. Thank you.
2, actually. Couple of cusk eels. And an Oreo. And quite a few heterocarpus shrimp.
They got their own little oasis here.
Yeah, the heterocarpus, definitely saw quite a lot of those on the beta cameras. I think they were one of the most abundant things that we saw. I think there was one video frame where there was over 40. And we know now that they can actually aggregate in enormous groups of thousands. There was a recent paper that came out.
Good
Really?
for
From-
tennis.
Yeah, so there was a Gulf of Mexico
Ridge nav, 2
observation
meters,
from
180.
quite a while ago that just got published. by Steve Ross, and there's just pictures of the seafloor that are just
10 to south.
carpeted with thousands of heterocarpacents per
I have not seen that one.
possible spawning aggregation.
They also will eject bioluminescent fluid when scared.
Oh, can we look at this fish first? And then this weird orange stuff.
Okay, you can push in there, please.
Another small rat tail.
Swim right up to
Yeah,
the
that
orange
could be-
stuff for you.
Okay, whatever this is. Not entirely sure. It looks like a mushroom coral. It's like the most deformed mushroom coral I've ever seen.
Push in a bit more there if you want.
Yep, it's a mushroom coral.
Tilt up just a bit for me, Jade.
Copy.
With a snail eating it. Yeah, interesting.
Hmm.
Oh, look, this one's maybe regrowing?
Yep, got 3 or 4 polyps there.
Interesting. All right,
Strange.
thank you.
Okay. You're going.
Really getting back into the Primnoa-dominated environments here.
I'm ready for another 10, I think.
Bridge nav, 10 meters, 180.
Jellyfish. Here's your jellyfish, Amanda.
My dream. Oh, they're beautiful.
Fast moving jellyfish.
What kind is it, do you think?
That is
Wow.
one I don't
That's,
recognize. Like,
yeah, I don't
even.
recognize that either.
Oh my goodness.
That's interesting. The tentacles are so high up on the
Yeah.
on the bell.
They're pretty.
That's
Whoa,
different.
Whoa.
that's cool.
Some type of narcomedusa?
Hello. It's gorgeous.
Well, that'll be one to send to Dougal.
It's like super transparent.
Super active too.
Yeah, my best guess is some kind of narcomedusa, but I am by no means an expert when it comes to jellyfish.
was so cool. Thank you for letting me know about that.
Super abundant Oreos. I'm definitely really excited to analyze this data later and look at maybe what's causing the distribution of the fishes we see. If some of them prefer these high slope areas, high rugosity areas, high coral areas. It's been a very fishy cruise.
Looks like we're getting over the top here.
Yeah, we're good for another 10.
Bridge nav, 10 meters, 180.
Let's not strafe the top for a minute here, maybe.
Due south.
There's another Chaseon microniscus crab in the corner.
Nice little field of Calyptrophora. Big urchin.
Can we zoom on the urchin?
Sure. I'll get a little closer.
Saw some of these in the beta camera. Couldn't get a good ID on them.
Another Chaseon way back in the back. Big one.
Okay, can push in there.
We're into the dendrophilid corals too.
I think that should be good. Thank you.
Okay, what are we doing? I'm gonna slide to my left here for a minute. Where are we going after this? Up and over?
We're gonna start going to the west. Just follow along the ridge once we get up there.
Roger.
We still got maybe a little ways to go.
I'd say we're pretty much there.
Yeah.
Yeah. Atlantis 12 meters above me and see nothing but blue water.
All right.
Given that the current seems to be coming out of the south, I would be interested in pushing to the south a little bit further and seeing what's just over the ridge. And actually, we need to look at that, please.
Ooh, another Tomoptrid worm, it looks like.
Something in the water column there?
Yep, something in the water column right there.
Yeah, I missed the last one.
It's a worm?
This is a worm. It's a type of polychaete, a Tomoptritive. Sometimes called a gossamer worm.
Ooh,
It's pretty.
that's so cool.
Oh, what did they eat? Oh. Whoa. Wow.
There's a really iconic shot of one of these in the original Blue Planet series that I remember seeing as a kid and being like, what is that?
Oh, that's amazing. How do you feel seeing it now?
Pretty cool.
Yeah?
I want to know what the tail of it is. Like what those little...
Tail's bioluminescent, isn't it?
Like, are those little white dots, are those photophores or something?
I
have no idea.
This worm is known for being very bioluminescent, but I forget exactly what part of it is.
And for those of you who are thinking to yourselves, why don't they just turn the lights off and see it?
We drop the lasers, though.
If only we could do that. Unfortunately, to film bioluminescence, you actually need ultra low light, special low light cameras.
Turn the lasers
so
off.
If we just turned Hercules' lights off, we would not really be able to see it too well, unfortunately. But starlight camera technology is improving and getting cheaper, and hopefully those types of cameras will be placed on more ROVs in the future.
Yeah. What's happening?
That was epic flying, Dan.
Okay, where do we go?
Keep going south to the ridge.
Came up quite a bit there, 20 meters.
We got this field of Calyptrophora here, and you can see how they're all lined up more or less parallel with each other, perpendicular to the current.
Oh, never mind.
So this is one of those interesting questions. Can we look at this sponge, please?
Roger. You can put a lens on top of Herc for me, or just to the south is fine. Sorry, Brian. What sponge?
This sponge.
Bridge,
Roger.
22135.
Bring my head back around into it a little here.
Affirmative.
Nope, that's not the sponge we're looking for. Let's look at the crab instead.